Hello, everyone, and welcome to today's session, Mass Cytometry in Translational Research, Session 1. And I'm Susie Valdez of Labroots, and I will be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. Today's session includes three presenters, Dr. Shelley Herbrich, Dr. Kara Davis, and Dr. Bernd Bodenmiller. Each will present and have a Q&A following their individual presentations. I first introduce our first speaker, Dr. Shelley Herbrich, a postdoctoral fellow at the University of Texas MD Anderson Cancer Center, presenting on the topic of single mass cytometry, identifying mechanisms of resistance to immunotherapy in AML. For a complete biography of Dr. Herbrich, please visit the biography tab at the top of your screen. Dr. Herbrich, welcome. You may now begin your presentation.
Okay, so I would like to thank the organizers this year for inviting me to share a little bit of our work that we're doing down at MD Anderson, looking at mechanisms of resistance to immunotherapy in acute myeloid leukemia using single-cell mass cytometry. So normal hematopoiesis is classically described as a hierarchical structure with the rare hematopoietic stem cells, or HSCs, residing at the apex. And these HSCs are capable of both self-renewal as well as differentiating into more committed progenitor cells that will ultimately go on to give rise to the mature cells of both the myeloid and the lymphoid compartments. However, in acute myeloid leukemia, the accumulation of mutations and subsequent genomic instability in these early stem and myeloid progenitors can actually halt normal maturation, resulting in what we call the leukemia stem cells, or LSCs, that are capable of rapidly producing abnormal white blood cells and ultimately outcompeting the production of healthy myeloid as well as lymphoid cells. We know that the frontline standard chemotherapeutic agents are largely effective at eliminating the bulk of disease, driving patients into remission. However, most patients will go on to give rise to deadly relapse leukemia. So our lab is interested in understanding new therapeutics that will ultimately eradicate the complete leukemia stem cell disease for those patients, especially with refractory relapse disease. So since the approval of the first anti-CTLA-4 antibody, ipilimumab, in 2011 for a variety of solid tumors, there have been multiple immune checkpoint inhibitors, or ICIs, targeting molecules such as CTLA-4, PD-1, PD-L1, that have had widespread use and efficacy in solid tumor settings. However, the efficacy in the heme malignancy world remains far more limited, with ICIs receiving approval in only certain subsets, including Hodgkin's lymphoma and certain B-cell lymphomas. To date, there have been no immune checkpoint inhibitors that have received approval for leukemia. However, we do appreciate the critical role that B-cell activity plays in the control of AML after it was observed over 40 years ago that the graft versus leukemia effect associated with allogeneic stem cell transplant was effective at eliminating leukemic disease. So just a tiny background on immune checkpoint therapy using monoclonal antibodies. So there are a variety of co-stimulatory and co-inhibitory molecules that tightly regulate the response of T cells to both antigen-presenting and infected cells. And so this cohort, upon activation and stimulation through the MHC and T cell receptor, requires both co-stimulation through CD28, and also the most prevalent inhibitory molecules include the program death protein 1 as well as CTLA4. For this talk, we'll be discussing the interaction of PD1 and its cognate ligands PDL1 and PDL2. So there are now multiple monoclonal antibodies in trial for blocking either PD1, PDL1, or PDL2. And the mechanism is such that blocking the interaction between the PD1 and the PDL1 expressed on the tumor cells removes the inhibitory blockade and allows the T cell to go about its effector function of destroying the tumor cell. So there are multiple clinical trials now introducing ICIs into the AML setting, most of which are in combination with hypomethylating agents. And this is actually motivated by some research done at MD Anderson in 2014, where it was shown that hypomethylating agents could actually increase the expression of PDL1, PDL2, and PD1 in a variety of myeloid malignancies. And the data here actually demonstrates that over the course of treatment, it was patients that were resistant to hypomethylating agents had higher expression of both PDL1 and PDL2. It was also demonstrated that the PD1, PDL1 signaling axis has profound anti-leukemic response. So in a PDX model of acute myeloid leukemia, they found that treatment with an anti-PDL1 antibody significantly improved overall survival of these mice. And lastly, they have now achieved durable clinical responses in a hypomethylating agent PD1 inhibitor trial in acute myeloid leukemia. So the goal of our study was to use serially collected patient samples from refractory relapse AML patients undergoing therapy with a hypomethylating agent, azacitidine, in combination with the PD-L1 inhibitor avelumab for the treatment, as I mentioned, refractory relapse AML patient samples. But the goal was really with this study to use these samples and look at the single cell protein level to better understand the immune landscape of refractory relapse leukemias, as well as identify potential mechanisms of resistance in this setting. So the clinical trial is designed as such. It's a 28-day cycle where patients received the hypomethylating agent, azacitidine, for 7 consecutive days, in addition to 2 treatments of the anti-PD-L1 antibody on days 1 and day 14. For our study, we collected both peripheral blood and bone marrow at baseline, as well as serial measurements at the ends of cycles 1, 3, and 5. And after all of this processing, we were able to generate data, serially collected data for 9 of these patients. So in designing our CyTOF panel, what we were really interested in looking at is both the immune cells adjacent to our leukemias, and so we included phenotypic markers for both AML, as well as a more extensive T cell phenotyping panel. We also included certain markers for other known effector immune cells in the bone marrow and peripheral blood, including monocytes, NK cells, and B cells. And then we filled the remaining channels actually with a variety of immune checkpoints that we would have hypothesized be expressed on either the T cells or the adjacent AML cells. And since this is a very CyTOF-oriented crowd, I thought I would give just a little more specifics on how we chose to run these CyTOF samples. So all of the CyTOF samples were collected as part of the clinical trial and frozen between 2017 and 2018. So when we went back to evaluate these samples, we chose to take all samples from a given patient and run them in a bar-coded CyTOF batch. In order to control variability from batch to batch, we also use a pooled set of healthy PBMCs that we run with each as an internal standard. And we use the same antibody master mix for all of these, so to reduce any variability in the master mix preparation. And then lastly, we do all of our data pre-processing, which includes the removal of debris and dead cells and doublets using FlowJo. And then we perform UMAP analysis with Phenograph using CyTOF kit in RStudio. So using one of our AML patient samples, I'd just like to walk you through an example of how we went about analyzing this data. And so here what you can see is one AML patient sample at baseline from bone marrow. And you see a very nice separation between the CD3, both the CD4 positive T cells and CD8 positive T cells here at the bottom. That separates from this large mass of acute myeloid leukemia cells, and we actually detect acute myeloid leukemia as CD45 dim, and they also express markers of immature myeloid cells, including CD34 on the left and CD38 on the right. And what you'll notice here is that we identify a more stem-like leukemia in those that are CD34 positive, CD38 negative, in this cluster here to the left. So our original hypothesis was that these AML patient samples at baseline would express high abundance of PD-L1 on the blast cells, and that these would either be selectively eliminated over the course of treatment. However, what we saw in the majority of our patient samples upon first inspection was that at baseline we saw very little PD-L1 on the tumor cells. And so then we next hypothesized that maybe after a first cycle where they had been exposed to the hypomethylating agent, we would see an increase maybe of the PD-L1 in that context. However, as you walk through the same UMAP projection of this patient sample throughout the course of treatment, what you'll see is that PD-L1 expression does not change significantly after one cycle of treatment or after 3 cycles of treatment. And even after 5 cycles of treatment. And in fact, we saw no selective inhibition or regulation of PD-L1 in this context. So because we ran our experiment with a number of other checkpoints, we were able to go back and specifically look at the AML blasts from a variety of patients. So in this particular heat map, we're looking at the fraction of AML cells, or the percentage of AML cells that express a certain immune ligand across 8 different samples for which we had serial measurements here. And what we were surprised to see is that across the board, we saw very little PD-L1 expression in these refractory relapsed AML samples. However, we were surprised to find that its paired ligand, which is PD-L2, appears to have high expression across the board and across disease progression. So we looked both at the peripheral blood and baseline of for 6 of these for which we had both peripheral blood and bone marrow measurements. And what we found was that the PD-L2 was actually significantly higher both when looking at the AML in the peripheral blood as well as when looking at the AML resident in the bone marrow. And so we went back to the data and we looked through these patient samples to see PD-L2 expression over the course of treatment. So again, here you see in our more stem-like population, we have the highest expression of our PD-L2. And as we watch this patient progress over treatment, we see that the PD-L2 is upregulated after cycle 1, persists even through cycle 3 when the patient blast count is lower. And you'll notice here a small population that's beginning to emerge that is present when the patient is removed from trial due to non-response. And again, we see that the PD-L2 expression is highest at this point and is also present on a newly emerging clone of leukemia. So in a similar kind of heat map, we can look at not just the baseline expression or the baseline abundance of these other immune checkpoints, but what we see is actually here, the numbers in the heat map represent the percentage of blasts positive at baseline, and then we've color-coded this to say whether or not a patient in each column has increased by greater than 5%, stable, or decreased by more than 5%, the abundance of blasts with these particular immune checkpoints. And so what you can appreciate is that in most of the patients, we see that the majority of these other immune checkpoints are either stable or increasing over the course of treatment. And so the summary for this first part of the data is that PD-L2 is actually our most abundantly expressed immune checkpoint that we interrogated. And this may help to explain some of the discrepancy we see in the clinic in response to PD-1 inhibitors versus PD-L1 inhibitors. This also gives us the increased expression of some of these other immune checkpoints, including TIGIT, OX40, and TIM3, gives us a real appreciation to the complexity of immune control in the myeloid setting. And this is something that we are considering for future immunotherapy trials. So the other thing that we were able to do with this data is actually look specifically at the T-cell compartments. And for this, I'm using a different patient sample, but what we saw and what tended to be the trend is that in these refractory relapsed AML patient samples, when looking between peripheral blood and bone marrow at baseline, the majority of patients had significantly more T cells in the peripheral blood than when compared to the bone marrow, which is something that we've seen in other leukemia settings. But what was interesting is that in most of the patient samples, we could actually see an influx of T cells after one course of treatment with the hypomethylating agent and PD-L1. Whether it's due to the monoclonal antibody or the hypomethylating agent is unclear, but it's something that we would like to consider going forward. So we took the T-cell numbers and ratios and looked for anything that could be a possible predictor of response to the PD-L1 trial. And sorry about the strange thing there. But what we found was that of the number of patients, so of the 8 patients that we were able to interrogate in this manner, we saw that those that progressed had significantly more or had a significantly higher CD4 to CD8 ratio when compared to patients that had stable or disease reduction in response to treatment. And we could summarize this another way by looking at actual changes in blast count from cycle to cycle. So what you see on the right is the CD4 to CD8 ratio. So if there were more CD8 T cells, it will be positive. Here on the right, if there were more, if there are more CD8 positive T cells, it will be here to the left of 0. Whereas if there were more CD4 helper T cells, the T cell ratio would be above 0. And so we compared this to the relative change in the blast percentage as clinically defined, and we found a very strong correlation between these two markers, indicating that response to this HMA plus PD-L1 inhibitor requires that you have an abundance of CD8 effector T cells. Another thing that we were able to look at with this data and with our immunophenotyping of the T cells was actually T cell composition of refractory relapsed AML, which has not largely been investigated. So here what we're looking at are 3 normal bone marrows run with the same CyTOF panel, along with our 9 AML bone marrows for which we had baseline measurements. And what we see here is that both the CD4 and CD8 composition are significantly different in the refractory relapsed leukemia when compared to the normal bone marrow. And notably, we saw very, very few terminally differentiated and naive T cells in the AML bone marrows when compared to the normal bone marrows. We could also look at this T cell composition over time. So again, this is one AML patient sample that we tracked from bone marrow at baseline to C1 to C3 to C5. And what we found, interestingly, in both the CD4 and CD8 T cells, was the elimination of the terminally differentiated, as well as in the CD4 case, the naive T cells over the course of treatment. Again, whether this is due to the hypomethylating agent or the anti-PD-L1 is unclear, but it's something that warrants further investigation. In the second sample, we see a similar trend with the reduction of naive and terminally differentiated cells and the expansion of effector memory T cells. So the summary of the second portion of this data is that using the same clinical trial data set, we were able to make a few claims about the T cells of patients with refractory relapsed AML and determine that CD4 to CD8 ratio was the most important predictor that we had of response to the HMA plus PD-L1 inhibition. We also found that these refractory relapse samples had significantly more memory T cells and far fewer naive and terminally differentiated CD4 and CD8 T cells, both pre-treatment and over the course of treatment when compared to healthy controls. Again, whether these are specific to refractory relapse when compared to de novo AML or when compared to age-matched is, again, under investigation, but worth considering as we continue to expand these immunotherapy trials into AML. And so with that, I would just like to thank the Konopleva Lab, which helped considerably in the collection and processing of all of these clinical trial samples. The MD Anderson Leukemia Department, this trial was headed by Naval Daver, as well as our MD Anderson Flow Corps, especially Duncan and Angelique, who are our CyTOF gurus and who run all of these for us. So with that, I will take any questions.
All right.
Thank you, Dr. Herbrich, for that informative presentation. And unfortunately, Dr. Herbrich could not join us for the live Q&A. Any of the questions submitted today will be provided offline. So go ahead and submit those and she will be sure to answer them. But we do want to thank her for her time today and her important research. I now present our next speaker, Dr. Kara Davis, an assistant professor of pediatrics at the Stanford Child Health Research Institute, presenting on the topic of understanding CD19 negative relapse following CAR T therapies in acute lymphoblastic leukemia. For complete biography on Dr. Davis, please visit the biography tab at the top of the screen. Dr. Davis, welcome. You may now begin your presentation.
Good morning, everyone. Thank you so much for the opportunity to present an ongoing project in our lab, and happy to be discussing it with you today. We're going to talk today about our work using mass cytometry to help understand CD19 negative relapse in patients who suffer these relapses following CAR T immunotherapies for acute lymphoblastic leukemia. And I want to right away give attention to the fact that this work has all been led by a fantastic postdoc in the group, Pablo Domizzi, and give him credit where credit is due. So one note historically is that 100 years ago, childhood leukemia was universally fatal. This is a case report from a doctor in Birmingham who reported a few children who presented with large organs and high white blood cell counts and invariably succumbed to their disease within only a couple of months. But B-cell childhood leukemia, which is the most common type of cancer in children, is really a poster child for success in cancer treatment. These Kaplan-Meier curves I'm showing you are from the studies consecutively at St. Jude Children's Research Hospital, which really shows the progress from the 1960s in which less than 10% of children survived up until now, actually even a decade ago, where for many children we can quote survival and long-term survival over 90%. However, one of the main challenges that we still face with these patients is relapse leukemia, and about 15 to 20% of children still relapse. And even though we have had this fantastic new option for these patients, which is CAR T cells, which are T cells that are engineered to recognize a protein on leukemia cells, we still have a significant amount of patients that fail these therapies. This Kaplan-Meier curve is from the sort of pivotal trial of tisagenlecleucel, which is the Novartis CD19 CAR T cell, and this is what led to the approval by the FDA for this first CAR T cell therapy. And we participated in this study at Stanford, and one of the things that was as fantastic as it is in getting patients into remission, you can see in the black curve, which is event-free survival, that almost half of patients did have an event, and most of these were relapses, and the majority of which were patients who relapsed in which their leukemia no longer expressed the CAR T cell target of CD19, so a CD19 negative relapse. So in this project, we really wanted to focus on failure with CAR T cells and try to understand who will have CD19 negative relapses, and if we can understand at the time that the patient receives the CAR T cells, are there cells within that mixture, billions of cells, that are more likely to become CD19 negative and mediate a relapse. And this is really in line with what we try to do in my lab, which is using mass cytometry as a way to look at primary patient samples and understand cell populations that are associated with clinical endpoints. So we already know a bit about CD19 negative relapse in the face of targeted therapy, both CAR T cells and blinatumomab, which is another bispecific antibody, or a BiTE antibody actually, that targets CD19 positive cells. So there's been description of frameshift mutations in the CD19 extracellular domain that then makes it difficult for the T cell to recognize CD19. There's been description of transdifferentiation of cells to a myeloid lineage, so that with pressure on CD19, the cells altogether can switch to looking more myeloid. There's been reports of disruption of CD19 trafficking to the membrane such that the protein gets stuck intracellularly, and this actually can be mediated through also a chaperone protein for CD19, which is CD81. And then there's also been a description of intracellular isoforms or splice isoforms of CD19 that, again, either makes the protein not express the part of the protein that is recognized by the CAR T cell, or potentially that involves, again, localization within the cell. And this has been described in 2 different papers also with intron retention. So from our standpoint, we wanted to look at the pre-CAR T cell sample, and we had 2 hypotheses to try to explain CD19 negative relapse. One that we call immune pressure adaptation, meaning that you have a uniformly CD19 positive cell population going into CAR T cells. Under the pressure of the CAR, you end up with CD19 negative cells through some mechanism, versus a contrary hypothesis, which is an immune pressure enrichment, meaning that within the majority of the cells that seem to express CD19 before you give the CAR T cell, that there is hiding within there some CD19 negative cells, that then those are the cells that can escape the pressure of the CAR T cell and grow out to recapitulate the leukemia. We started by taking some of the RNA sequencing data that was in Elena Sotillo's cancer discovery paper, which they had where they described the splice isoforms, and we just looked at immunoglobulin rearrangements within that data And here I'm showing you two patient examples, both looking at heavy chain on the top row and light chain rearrangements in those two patients. And we could see that there were shared sequences, both in the heavy and light chain in the pre-CAR sample and in the post-CAR sample at the relapse, suggesting that there was, some clones that were there at the beginning before the patient had even seen the pressure of the CAR and persisted to the relapse. Collaborating then with David Barrett and Steve Grupp at Children's Hospital of Philadelphia and University of Pennsylvania, we were able to take a look at initially at 4 paired samples from patients who had gotten CD19 CAR for relapsed ALL. These were all expanded in a mouse, so they're actually all xenograft-derived samples. And just by using our standard ALL panel with the CyTOF, we looked at 42 markers, and just doing an initial high-level look at the phenotype of these samples, we could see that they actually segregated based on their genetic or mechanism of CD19 loss from what we knew of the samples, such that a healthy bone marrow that was analyzed with them completely separates away from the rest of the leukemia samples, and we see that CHOP1, which is the patient that had the CD19 19 splice isoforms, both in the pre-CAR and post-CAR sample, separates phenotypically away from the 3 other patients that were known to have mutations in the CD19 gene. One of the tools that we use in our lab to look at leukemia samples is a developmental classifier in which we classify leukemia cells based on their most similar B cell population in the bone marrow. And when we applied our classifier to this data now to segregate these cells into subpopulations, interestingly, what we found was that at the time of CD19 loss at the relapse, which is shown in red, there was this significant enrichment in cells that classified as early non-B1 cells in our classification. So we were really lucky then to be able to expand our cohort of patient samples to try to see if this was something that was specific to this small pilot set, or if we saw a similar pattern when we looked at additional samples. And again, all of this is through collaboration with David and Steve at University of Pennsylvania. And here then we were able to look at patients who had complete remission after their CAR, patients who suffered a CD19 negative relapse, and for those patients we had 8 paired post-CAR relapse samples. We had a smaller group of patients who suffered a CD19 positive relapse, and for those we had 2 post-relapse samples. And then finally, we had 5 patients who received a 19-directed CAR but never had any response, and so were termed refractory. And for these, we had also some paired samples. So just looking at CD19 alone, we noted that there was no difference either in the percent of CD19 positive cells, especially in the pre-CAR sample, between these 4 groups of patients. And then if we look actually at the median level of CD19 in the cells, there was no difference between these groups, aside from the patients who went on to suffer a 19 negative relapse after their CAR, which there was obviously a statistically significant difference in both the number of, or the percentage of 19 positive cells and the level of 19 on the cells. But then when we did our developmental classification in these different groups, we found that in the patients who went on to a complete remission, that they tended to have a very typical expansion in what we call pro-B2 cells compared to the other groups. And here I'm highlighting patients who went on to complete remission versus those that had a 19 positive relapse. Here's what it looks like for patients who had a complete remission versus those who had a 19 negative relapse. And here's what it looks like for patients who had a complete remission versus who were refractory. So in each case where patients either were refractory or suffered a relapse, there were less cells in this Pro-B2 population, although this only reached statistical significance when comparing to the refractory patients. When we went and looked specifically now at the patients who had a 19 negative relapse and looked at the paired samples now with more patients, again, we saw this enrichment in the early non-B cell population, which was, again, significant compared to the pre-CAR sample. And just to drill down into how our developmental classifier works, we use this by taking healthy bone marrow controls that we always include in our experiments. The healthy bone marrows are gated based on what we know is the progression of B cell development, and then each healthy population is assigned a developmental barcode of 11 different proteins. In this case, though, we always use CD19 because CD19 is a known important marker of B cell-ness. But in this case, we asked the question about whether or not what was happening when these cells don't have CD19. So we asked what would happen if we excluded CD19 from the classifier, but used the remaining markers. And this I'm showing here is healthy bone marrow, our healthy bone marrow controls. And when we actually include in the blue on the left, CD19 in the classifier, you can see that there's perfect agreement of where cells are classified, versus if we exclude it, again, there's very little impact on where cells are classified, whether or not 19 is there or not in our classification. But this was not the case with the leukemia cells. And in fact, what we ended up seeing was that in the leukemia samples without CD19, we found these cells that we call Pro-B2 discordant cells. And so what that means is that when we include CD19 in the classification, these cells are called Pro-B2 cells. But when we exclude it from the classification, they end up being called early non-B1 cells, which sort of explains why we saw this enrichment in that population in the CD19 negative relapse samples. However, within these patient samples as well, there were some cells that with or without 19 maintained their classification as Pro-B2 cells. So really there was a subset of these cells that changed sort of what they looked like, whether or not we used 19 to define them. We actually went to another data set that we had in the lab, which is de novo ALL. So these are patients who are newly diagnosed and have never had CAR T cells. And we asked the same question about, can we find these discordant Pro-B2 cells by classifying these patient cells with or without CD19 in the classifier. And out of our 22 samples, we had 17 of them that had more than 100 cells classified as Pro-B2. And here this bar graph is showing you the percentage of those Pro-B2 cells that would be considered discordant if we excluded CD19 from the classifier. So this told us that these discordant Pro-B2 cells are not just specific in some way to patients who are receiving CAR T cells or even necessarily to relapsed ALL, but they appear to exist in both conditions. And here I can show you a summary of the frequency of Pro-B2 cells within all of our groups. So they certainly were present in patients who went into complete remission with CAR. They were present in patients who had a 19 negative relapse, as well as refractory or 19 positive patients and de novo ALL as well. But we did not see them in healthy bone marrow. So when we started to compare on the protein level, the differences in the proteins that are expressed between all of the different combinations here, so Pro-B2 versus early non-B cells, Pro-B2 discordant versus Pro-B2 cells, and then Pro-B2 discordant versus early non-B cells, we could see quite a lot of proteins that were differentially expressed. And these included a lot of interesting players, I think, in sort of B-cell identity, including PAX5 and Icaros and PU1, so transcription factors that help regulate cell identity, as well as other CAR targets such as CD22 or, for example, CD20, which is not a widely used CAR target, but there are things in development to target CD20 as well as other monoclonal antibody agents. When we started to drill down a little bit more into then, doing what we love to do with the CyTOF, which is to be able to look at multiple features within these individual cells or in these individual populations, we found that in the patients who are going to have a 19 negative relapse, that these ProB2 discordant cells do have some unique features in comparison here to patients who go on to have a complete remission. So for example, as I had previously shown you and you would expect, the discordant ProB2 cells have really a very slight difference in CD19 in the pre-CAR sample. However, they do have differences in PAX5, in phospho-NF-kappa B, and in Ikaros, and that the patients who go on to have a 19 negative relapse have lower levels of these proteins in their pre-CAR sample in the ProB2 discordant cells. And when you start to look at them combinatorially, there's differences in between these cells where the patients that go on to a negative relapse will have lower levels of PAX5 and Ikaros in the pre-CAR sample. And this is provocative in the standpoint that CD19 is a known target of PAX5, so it makes me wonder about whether or not these are cells that are somewhat poised to already suffer 19 negative relapse because they're wired differently to enable either 19 to turn off under pressure. We wanted to understand a little bit also if there was a CD19 negative early non-B cell in healthy bone marrow, since this was what defines these early non-B cell populations that we were seeing with the classifier. So Pablo took a look at 4 different healthy bone marrows, ran the classifier, and took the cells that were identified as early non-B cells and performed an unsupervised clustering. And within that one population that through classification we call early non-B cells, we found 3 clusters. If we looked at these 3 clusters, we could see that within them they were 19 negative, but there was, here shown in blue, cluster 3, a population there within the 4 marrows that expressed immunoglobulin heavy chain, or IgM, on the surface and had HLA-DR, suggesting that this could be, sort of a cognate population in healthy bone marrow that could lead to CD19 negative relapse in these patients with leukemia. So in order to follow all this up, one of the things that we've now started to look at is looking more closely at these samples using sequence-based cytometry with whole transcriptome analysis in our ProB2 discordant cells, in actually all of these leukemia cells, and using, using AbSeq to be able to identify both our ProB2 cells, our early non-B cells, but also then look at the whole transcriptome within these cells. So what we did is we did a total of 9 samples, so 3 patients who before got their CAR and who ended up having a complete response, 3 patients who ended up having CD negative relapse, and then we have those paired from before and after, so pre-CAR and post the negative relapse. And as well, we have a healthy bone marrow that's been included on all of these cartridges. So similarly to what we've done previously with CyTOF data by projecting ALL cells into healthy bone marrow space on the protein level, we're starting to doing here on the tryptome level. So here's a UMAP showing you on the left-hand side what the healthy marrow controls look like through clustering and UMAP visualization. We get 17 different clusters in the healthy bone marrow space, whereas on the right side is where leukemias cluster into this space. And we found that primarily they occupied cluster 6, cluster 14, cluster 7, and cluster 9. And I'll show you this a little more concisely on the next slide. But in particular, looking at the gene expression, cluster 6 BB are discordant cells. And I will give you a little bit of an idea why I'm coming forward, whereas cluster 14 and cluster 7 look like they are very early pro-B cells and cluster 9 pre-B cells based on their gene expression. So here's the same sort of a line graph that showed you with the developmental classifier with these samples, though in this case now the cluster ID is on the x-axis, so it doesn't necessarily correlate with a developmental stage across the x. But as I mentioned, in all these samples, including the patients who go on radar, most of the cells sit in cluster 6 or cluster 14. And you can see within the patient on the top row have pre and post CAR samples. There are some swaps in frequency that happen between the pre and CAR sample, but for the most part, they still occupied 2 specific clusters. So looking a little bit more deeply in cluster 6, we are asking the question, are these potentially what we had previously identified as discordant pro-B cells? Pablo took the top 100 cells from all of the patients that we looked at and did some differential analysis to the gene expression, and you can see that there are both upregulated and downregulated sets of genes that separate these 2 clinical statuses. Some of them include things we would have expected, such as TT, as well as HLA, genes that regulate HLA and regulate the immunoglobulin heavy chain, which I have here highlighted in red. So this is all ongoing work, so to be continued. So stay tuned as we continue to get in analyzing this additional single cell data and overlaying it with CyTOF data that we had. We're really excited about integrating these 2 data types and using them to inform each other as we go back to potentially look at more patient samples. So I apologize if this is cut off on the top, but to summarize our proposed models, where is the source of these CD18 resistant cells? What we think may be happening is that this immune pressure adaptation, where there could be cells in what they are feeding, they are all CD19 positive or appear to be lower pressure of CAR, but in fact, there are these cells that are pre-2 discordant cells that initially express 19 but are more prone to losing 19 under pressure. And so there actually may be sort of a merger of these 2 models where you may have both things happening at the time. And so with that, I'm going to wrap up. I'm going to say thank you to all the people who've been involved in this. But of course, like I mentioned, the work has been led by Pablo Domizzi, and then of course in collaboration with Dave Barrett and Steve Grupp at University of Pennsylvania, with support and samples from Charles Mulligan and lots of input from Crystal Makel, and primarily supported by the D Foundation financially. So thank you so much for the opportunity to talk with you about this, and I'm happy to take questions.
Thank you, Dr. Davis, for that informative presentation. We will now start the Q&A portion of the webinar. If you have any questions you want to ask, please just click on that Ask a Question box located on the far left of your screen. So it looks like we already have some great questions coming in. Dr. Davis, have you applied the same analysis to patients receiving the targeted therapies in blinatumomab or CD22 CARs? Oops, it looks like we cannot hear you. You might have muted yourself on that end. If you go to the bottom of the screen where we can see each other, right where there's that little microphone, if you click on it, you can turn it to the correct microphone. Maybe you accidentally switched it to the wrong one. Or if it's is it red? Maybe it has a line through it and you want to unmute. Uh-oh. If you want to, if it still doesn't work, you can quickly jump out and then come back in. That could work as well. And I want to thank our audience members. We do have some great questions coming in. We want to encourage you to continue to add those questions. This is a great workshop, and we want everyone to participate. So we'll just give, these technical problems happen on occasion. We all know, don't worry. You can either click on that source at the bottom of the picture. It looks like she's gonna log out and log right back in. And again, thank you so much for all of your patience. I'm sure everyone has dealt with these kind of technological issues, especially throughout this pandemic. So thank you.
Okay, can you hear me now, Susie?
We can.
Yay. Sorry about that.
Thank you to our audience members. And I do want to just add that encourage people to join in at the Fluidigm booths in the exhibit hall too. This has been a great opportunity, and Fluidigm has been awesome in this whole experience. So thank them. So Kara, let's go back to these great questions, and we already have some coming in, and I know we'll get some more. Our first questions was, have you applied that same analysis to patients receiving other targeted therapies like the and I apologize for not saying it right, Biotunomab or CD22 CARs?
Yeah, that's a mouthful, that drug.
Yeah.
Anyway, I wanted to say thank you, Susie. Thanks for having me to the organizers here, and good morning or good afternoon to everyone joining us today. We have not directly applied these same analyses to patients receiving blinatumomab. Blinatumomab is the bispecific or BiTE antibody that also targets CD19 and has been really another exciting new agent for leukemia. But we are very enthusiastic and are working actually collaboratively to try to obtain some patient samples from patients who've received blinatumomab. We have actually, though, really been excited to been able to take a look at some paired samples from patients who received a CAR targeting CD22, which is an alternative target in ALL and other B-cell malignancies like lymphoma. So we have a collaboration with Nirali Shah at the National Cancer Institute, who had the first clinical trial for CD22 in the US. And then we also now actually run a CD22 trial at Stanford. So we've taken a look at some of those samples as well. That data analysis is certainly a little bit more immature, but we're certainly looking to integrate some of the observations we're making with the CD19 along with the CD22 samples, because we actually also have observed clinically that after CD22 targeted CAR, it is very common for patients to drop the level of CD22 when they relapse. So it's an antigen that almost seems like it is even more susceptible to pressure. So stay tuned.
Thank you so much. And are these types of studies being used in investigating other types of blood neoplasms with other therapies?
I mean, absolutely. I think I know in my group we have multiple projects going on looking at both AML and ALL with other therapies or other sort of emerging therapies. We've already looked at BCL2 inhibitors, which is a really exciting drug that's being applied both in ALL and AML. And of course, we heard in the previous presentation from Shelley about, looking at these leukemias in response to checkpoint inhibition. And then the advantage there also being able to look at the immune system from the same patient samples, which really is advantageous when you're trying to do this sort of translational work where you have potentially limited cell numbers from primary patient samples and being able to leverage looking at many different cell populations from one precious sample.
Thank you so much. Now, one of our audience members want to know, Can you speak to the advantages of using mass cytometry and AbSeq in your research?
So that is something I'm pretty excited about in the sense that we oftentimes, have identified these cell populations of interest using mass cytometry, and then to have the opportunity to sort of use that to inform what cell populations we want to go after and even get more information on them. I think we're all sort of hungry for parameters. And of course, with the CyTOF, we can do amazing things. But then, of course, doing sort of unsupervised transcriptomic analysis, we get even more parameters. And I'm really excited about the opportunity to start to merge and overlay these data types so that we can get even more precise understanding of these cell populations. Because one of the things that sort of continually amazes me with doing this work is the incredible diversity of cells that we have in our bone marrow, which is primarily where we're working. But of course, this probably applies to all of our body. And so being able to understand those different cell types and their sort of propensities will help us hopefully learn how to treat patients better, down the road.
Thank you so much. Now, is there any utilization in the near future for these therapies in non-cancer diseases with, like, let's say, an immune dysfunction, example, liver pathologies?
Yeah, so CAR T cells are actually being used for infection as well, and particularly viral infections following solid organ transplant, for example, or bone marrow transplant. And so there also certainly is opportunity just to understand and potentially target cell populations that are a problem when in either rheumatologic diseases or autoimmune diseases. So I think there is an opportunity for using them both there's already ongoing work, but again, further expansion of this work into other areas.
Thank you so much. And again, I want to thank our audience for these great questions coming in. Are these types of studies being used in investigating other types of blood neoplasms, disease in general?
Yeah, so I think that exactly, that there are opportunities also to look at solid tumors or, solid tissues, we'll put it that way, both from the standpoint of looking at immune infiltrate, but also looking at the tumor tissue themselves. There's already been some published work doing tissue dissociation to be able to take solid tumors and look at individual cells, which is, of course, one of the, the real power of these technologies of CyTOF. So I think, those things are coming, and there's, protocols and experimental approaches to address, and be able to expand the reach of the technology.
Thank you so much. Now, do you potentially see a role for using mass cytometry for assessing minimal residual disease in general?
Yeah, so great question. We actually have a whole nother project working on that, led by another postdoc in the lab. And I'm not sure that I see a CyTOF in every clinical lab, but certainly we are learning that you can, if you assay a lot of different proteins and look at a lot of different markers, you can potentially identify the best set of markers to look at, for example, minimal residual disease. And so, I think that there absolutely is a role. We know that both with the ability to look at so many proteins and sort of the analysis techniques that we are really nicely able to find abnormal cells, which for us clinically helps us guide treatment decisions.
Dr. Davis, thank you so much. Would you like to provide any additional remarks before we move to our next speaker?
Sure. I think I wanted to say, again, thanks for the opportunity to be here with you all this morning and to talk about our work. And certainly I'm very interested to stay on and listen to my colleague, Dr. Bodenmiller, who's following in his very relevant work to to our unusual times. But thanks everyone for being here with us today.
And thank you again, Dr. Davis, and thank you for your time and for your important research. I now want to move to our last and wonderful speaker, Dr. Bernd Bodenmiller, Professor of Quantitative Biology at the Department of Quantitative Biomedicine at the University of Zurich, presenting on the topic of mass cytometry reveals distinct immune signature marking progression from mild to severe COVID-19. For complete biography of Dr. Bodenmiller, please visit the biography tab at the top of your screen. Dr. Bodenmiller, welcome. You may now begin your presentation, sir.
Thanks for the nice introduction. And also I would like to thank the organizers of the meeting for giving me the opportunity to talk about our recent work on the immunoprofiling of peripheral blood of mild and severe COVID-19 patients. As you all know, we have currently an ongoing worldwide pandemic caused by the SARS-CoV-2 virus. Over 29 million people already have been infected with this virus, and nearly 1 million people have died of it. Here you see a world map that shows which countries have been mainly affected. And in Europe, we are through the first wave, but in most countries, there's already, again, the ramp up of cases in some countries like France, actually steeply, and also in Spain, where we already are in the midst of a second wave. In COVID-19, patients can be asymptomatic. Many have only mild symptoms, but some of them show severe symptoms, and about 5% are ultimately critical cases. And overall, we have a fairly high mortality in this disease, which is estimated to be between 0.5 to 1% of all cases. And we aimed to generate high-dimensional single-cell proteomic data to help us understand the key differences between the mild and the severe disease, to ultimately identify early on patients at risk, and of course also to help understand the underlying biology of the disease. And to address this, we performed mass cytometry and targeted proteomic measurements to characterize a cohort of mild and severe COVID-19 patients. And those samples were collected in 4 different hospitals in Zurich, Switzerland. As a side note, the results of our work is described in 2 manuscripts that you can find on bioarchives under those 2 links. One focusing on the innate compartment, and the second one focusing on T cells. In the presentation today, I will show you parts of both of these manuscripts and analyses. In this data set that I will show you today, 66 COVID patients are included. These were sampled at different time points after symptom onset. To the left, you see number of patients. X-axis is the time after symptom onset. And you can also see in a color code whether those patients had mild disease or a severe disease or even acute respiratory distress syndrome. As it's known for COVID-19, among the severe cases, men are actually overrepresented. We also found, as others, a strong relationship with age for the disease severity that is shown here to the right, where the most severe cases are among the oldest patients of the cohort. Also, if we look at the hospitalization, we see a strong relationship to the age of the patient. And also, when we look more finely grained at the different disease features, we again see, as expected, a strong relationship to the age of this patient. We then took all of these patient samples and in addition had 22 healthy donors, and we analyzed the PBMCs using an antibody panel for the innate cells as well as for the adaptive immune cells. And each panel had contained 40 antibodies, and we analyzed all of those cells then by mass cytometry, and overall we acquired about 6 million cells in 2 batches using a 60-well barcoding scheme. In parallel, we used the serum of all of these patients, and we did cytokine profiling using the so-called O-link approach. In the first step, then we identified the main immune cell subsets using a random forest classifier. To the upper left, you can see a T-SNE map that shows the different main immune cell phenotypes, including CD8, CD4 T cells, B cells, plasma cells, dendritic cells, monocytes, NK cells, basophils, neutrophils, etc. To the lower left, you see a heat map. Then the x-axis, you see the different markers that we analyzed. On the y-axis, the different phenotypes and the expression of the markers from blue lowest to red highest level. We then plotted the cell type frequencies, which are shown to the left, versus the disease severity. And here we found, and this other people also found while we actually analyzed our data, that there's a depletion of CD8 T cells among the severe patients, but that there's an increase of the monocytes also among the severe patients. And this then led us to analyze in more depth the monocytes as well as the T cells, or the myeloid compartment and the T cells. And when we then looked closer at the myeloid compartment, I first want to show you this T-SNE map here that is colored by the disease grade. And in green, you see cells derived from healthy patient samples, blue from patient samples from patients that had mild disease, and red from patients that had a severe disease. And please note that there's a region that is exclusively occupied by cells from patients with mild and severe disease. Now here to the right, the T-SNE map colored by the myeloid specific markers, again from blue lowest to red highest, normalized intensity. And then it was striking to see that CD169 showed a very strong relationship to the region where we had the cells derived from samples of patients with mild and severe disease, and also other markers such as CD64 in fact showed some relationship to the disease state. And this clearly showed us that CD169 was strongly upregulated in the myeloid compartment in a subset of COVID-19 patients. We then used phenograph to determine the main monocyte phenotype clusters. These are shown here on the heat map. Again, on the x-axis, you see the markers that we measured. The color scale is the median scale expression from blue lowest to red highest. And this enabled us to determine the non-classical and intermediate monocytes that express CD16. And here expressing CD169 activated monocytes, and down here expressing CD14, but not expressing CD16, the classical monocytes. In the next step, then we wanted to know how these monocytes, these activated monocytes, where they might have been originated from. So we analyzed the putative ontogenic relationship between the different monocyte clusters, and to do so, we used diffusion mapping. And the diffusion mapping actually showed us that these activated monocytes could be derived from these classical monocytes, but also from those non-classical monocytes. And in HIV, it actually had already been described that the CD169 positive monocytes can be found both on CD16 positive and CD16 negative monocytes. So clearly could be from the classical and the non-classical compartment. In the next step, we wanted to see which of these monocyte phenotype clusters are associated to the disease states of our cohort. For that purpose, we made a stacked histogram plot where for the different monocyte clusters, the color scale is shown here to the right, where we plot the proportion for every individual patient of the different monocyte clusters. And the x-axis is showing all of the different samples. Now, just looking at this stacked histogram, I think it's very clear that there are three groups. This is group here to the left, where we mainly have the activated monocytes. Now, here on the top, you can see the disease severity. And you can also see that for those patients and samples where we mainly have these activated monocytes, we typically find patients that had a mild or a severe disease. Then there's a second group here in the center, where we have many classical monocytes, and these are associated mainly with the healthy donors. Then there's a third group that is shown here to the right, where we have higher proportions of a subset, for example, it's 6 classical monocytes, and these are associated with severe disease. So then we analyze this more systematically using a box plots which represent cluster frequencies across those patient groups. Now here on the top, you see the classical monocytes, here to the left, the non-classical monocytes, and here to the bottom right, you see the activated monocytes. And you can now see that for these activated monocytes, you have higher frequency for the patients with mild disease, while for the classical monocytes and here for the non-classical monocytes, you can see that their frequency is actually reduced for the patients with a mild disease. In the next step, we then wanted to analyze the pseudodynamic of these different monocyte clusters. And here we could exploit that we sampled the patients at different time points after symptom onset. And so here on the x-axis, then you have the time after symptom onset, the y-axis, the frequency. And several things then become clear when we look now at these clusters of the classical monocytes. The first, like this cluster M1, M2, and M3, they're depleted already at the early time points of the onset of symptoms. But over time, at least for the severe and also for the mild patients, there's a slow recovery. These groups M4 and M6, they actually appear to be transient, that there's an increase in the frequency over time, and then also they decrease again over time. And then for this M7, there was only a very weak relationship found over time. When we then looked at this non-classical monocytes and the activated monocytes, we could also see here very strong relationship in their frequency with time after symptom onset. So clearly there was an increased frequency of the non-classical monocytes in this more severe patients. That's the red line. You can see how there's a strong increase. While if you look now at the CD169 positive deactivated monocytes, you can see that early on after symptom onset, you have a high frequency, but then that is transient, and that then the frequency is actually reduced over time. So very often, monocyte development, homeostasis, and also the fight, I mean, these points are strongly interlinked with the cytokine and chemokine environment. So in order to probe the cytokine and chemokine environment, we use the O-link targeted inflammation assay. And so here you have two antibodies binding against the cytokine or chemokine. On the antibody, you have a DNA tag. Then when both antibodies can bind and they are close enough, then you can do a proximity extension assay. And after the extension, you can do a pre-amplification, and then you can do a qPCR to quantify the amount of a chemokine or cytokine in your sample. And so we then compared which of those cytokines, chemokines are differentially expressed, comparing the healthy versus disease severe cases. And you can clearly see how pro-inflammatory cytokines such as IL-6, TNF, IL-18, or interferon gamma are actually significantly upregulated compared to the healthy controls. And overall, we analyzed 92 inflammation markers. So also here, then we performed a pseudo-dynamic or pseudo-time analysis to look at the pseudo-dynamic changes in the cytokine expression upon symptom onset, which again is shown or plotted on the x-axis, the time after symptom onset. And now the normalized protein expression is shown for the different markers that are shown here on the y-axis. And one can now clearly see that, for example, in the case of interferon gamma, early on, the high levels for the severe cases, but that it then fades over time. When we look at CCL3 or CCL4, we can see that there's an increase of the levels over time for severe patients. And also MCP2 has a similar behavior like interferon gamma in that it's high in the beginning, but then it fades over time. And also other markers showed some weaker trends. MCSF had high levels initially, and there we saw a slow decrease for the mild cases over time. We then wanted to see whether we can stratify the patients based on cellular and soluble factors. For this purpose, then we performed a principal component analysis. What is plotted here on the x-axis, the first dimension, on the y-axis, the second dimension. And it's very clear that we can separate out, shown in circles, the healthy cases. We can separate fairly well the mild cases that are shown in triangles. And we can also separate, using PCA, the severe cases that are here in the region at the bottom. Then we performed biplotting to find which of the monocyte clusters are associated with healthy, mild, and severe disease. And as you already have seen before on this large plots that I've shown you, the stacked histograms, then that this classical monocyte clusters M1, M2, and M3, they're associated with the healthy samples, or that up here, this M10 and M12, they're associated with a mild disease, and that down here, the M4 and M6 classical monocytes, they're actually associated with severe disease. And this really showed us that the combination of cytokines and cell clusters define the different groups of the patients. We then more systematically analyzed the relationship between the monocyte clusters and the chemokines and cytokines. And therefore, we performed a correlation analysis between, in this case, the M12 activated monocyte cluster and the cytokine levels. And here we saw a strong relationship between this cluster to MCSF, to MCP2, and also to interferon gamma. And we saw some trends also for other cytokines. We then also colored the patients in relation to the time after symptom onset. And we can see here that those patients that were analyzed early on are driving this relationship. And we can also see up here that there's an early stage signature that then shows us this association between the CD169 clusters 12, that's the cluster we looked at, and MCSF, MCP2, and interferon gamma. And also we see a relationship more to the mild cases than to the severe cases. When we also look at other clusters, M8 and M9, these are non-classical monocytes. Here we did the same analysis. And here we see for cluster 8 a relationship to CCL3 and CCL4. And here the relationship is driven by patients that had a severe disease. And the same is true for cluster 9. And if we now also color these plots according to the time after symptom onset, now we can see that it's driven by samples that were taken late after symptom onset. And this shows us that there's a late stage signature that identifies an association between this non-classical cluster M8 and M9 and CCL3 and CCL4. That means that indeed, what I stated initially, we could identify such relationships, and that we could identify signatures both for early stage and late stage of the disease, and for mild and the severe form. So then I just want to show you on a few slides some of the analyses we performed on the T cell side. And here it was striking to see that the naive CD8 positive T cells, that when we looked at the percentage versus disease severity, that there was a marked reduction. And also when we looked at the absolute cell count, we also saw a strong reduction. We then saw for the central memory one an expansion when we only looked at the percentage, but we did not really see that when we looked at the absolute cell count. And that really reveals the importance of assessing absolute cell counts and not only looking at frequencies or percentages of these different phenotypes. And overall, what we could see here is a pronounced reduction of naive T cells and overall a T cell lymphopenia. And so we at this point were wondering what actually accounts for this T cell lymphopenia. For that purpose, we first evaluated whether those cells become apoptotic, and therefore we made a plot of disease severity versus the expression of cleaved-PARP and cleaved-caspase-3. And indeed, we could see an elevation of those apoptotic markers for the among the severe patients. We then also wanted to know, and we hypothesized whether the T cell lymphopenia could also be caused by a T cell migration into inflamed tissues as another possible mechanism. And so here we looked at CXCR3 signaling, since that is primarily mediating T cell migration into inflamed lung parenchyma. And indeed, what we saw when we looked at the levels of CXCR3 ligands in the blood, CXCL9, CXCL10, CXCL11, then we could actually see a significant increase when we compared the severe levels to those of the mild and the healthy samples. And at the same time, we saw that there was a decreased CXCR3 expression on T cells in COVID expression on T cells in COVID-19 patients, and that is also shown down here. And this also indicates that in addition to apoptosis, that T cell migration into inflamed lung tissue might drive the T cell lymphopenia. So in case there's, due to this T cell lymphopenia, we hypothesized that there might be elevated levels of IL-7. So IL-7 is produced by stromal cells. Typically, IL-7 levels are fairly constant, but if there's a slight increase, and that's something that we could actually see here when we looked at the levels of IL-7 versus disease severity, we could see a trend of an increase of the levels, and especially when we looked at the time after symptom onset versus IL-7 levels. Also here we could actually see a trend that that there's a systemic IL-7 elevation. And IL-7 is important for T cell proliferation. And so here then we looked at the levels of the CD8 positive T cells and the levels of Ki67, which indicates proliferation. And here now we could see that indeed in the severe patients, we had higher levels of Ki67. And this indicates that IL-7 might induce CD8 T cell proliferation in severely affected patients. And we also saw that the CD8 T cell numbers, they increase with time. Again, when we plot the pseudo dynamics versus the cells of the CD8 T cells in blood, you could also see here a significant relationship, or more importantly, an increase with time in those severe patients. Then, at this point, we actually were wondering whether this homeostatic proliferation is associated to an increase in polyspecific memory T cells that might be produced using this IL-7 induced proliferation. And so we first checked the T cells from the COVID-19 patients, how well they were actually responsive on the level of blast formation to in vitro stimulation with different types of viruses that is shown here. And I think it's extremely obvious that the T cells from the patients that had severe disease showed a much lower blast formation and stimulation than what you can see in the healthy controls, and also from patients that only had a mild form of the disease. We then made another plot where we looked at the net stimulation versus time. So we again did the pseudo dynamic analysis, and on the y-axis you see the plot of the net stimulation. And here we can actually see an increased blast formation for the severe cases upon stimulation of the viruses shown here as a function of time. And ultimately, what this showed us is that the IL-7 dependent T cell proliferation is inducing a polyspecific T cell expansion. What I've also shown you is that T cell lymphopenia leads to an elevated IL-7 levels. At least we see trends that point towards this direction. And the underlying mechanism of the T cell lymphopenia is likely apoptosis, as well as the migration of the T cells into lung tissue. And on the innate compartment, I hope I could convince you that we have a clear innate signature in COVID-19 patients, that there's an early response that is characterized by a surge of CD169 positive monocytes and interferon gamma and MCP2, and that the severe patients present an increased amount of pro-inflammatory monocytes and elevated levels of CCL3, CCL4 at later time points. And with this, I greatly want to acknowledge first my team who analyzed those samples, and especially I greatly want to acknowledge Stefan's Herbrich. He really drove that project on our side. He did most of the data analysis, and he also drove the writing of this manuscript. And I also want to greatly acknowledge our collaborators at University Hospital of Zurich. First of all, Onur Boyman, and was an extremely fast, intense, and very nice, productive collaboration for the analysis of those patient samples, and also with Jacob Nielsen and the members of Onur's group, Yves Buchen, Sarah Adamo, and Carlo Cervia. And I also greatly want to acknowledge Andrea Jacobs, Suana Divapatam. They did most of the lab work and also joined in for the data analysis. And also I greatly want to acknowledge Natalie de Souza for contributing with the data analysis and manuscript writing. And finally, I want to acknowledge the Swiss National Science Foundation who is providing us the financial resources to conduct this work. With this, I'm done, and I'm happy to receive your questions.
Thank you, Dr. Bodenmiller, for that informative presentation. We will now start the Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. Dr. Bodenmiller, we have quite a few questions already coming in. We'll see what we can get in time today. Our first question is, what happens to the CD169 plus monocytes?
Okay, so that's a very good question, because we see that they're coming up and then they're disappearing again. And this activated CD169 monocytes, they could, for example, end up in the lung tissue. So they express CCR2. And we also see that in COVID-19 patients, CCL2 is upregulated. So they could, in the end, be recruited into the lung tissue and then ultimately also become macrophages. It has been shown in some mouse models also of COVID that this is the case. And that's our hypothesis, but we, of course, only analyzed the monocytes in PBMCs. So we do not know really what happens to them. And this would require additional analyses. That's in fact also a bit, I would say, a weakness of our study, that we did not analyze matched lung tissue, for example.
Thank you so much. Our next question comes from an audience member who actually asks a two-part question. How did you run these longitudinal samples? Was it at all some time point from one patient at a time with controls, like one full day? And then here showing comparison, for example, moderate or severe, our lab is determining the time of how we run CyTOF samples from these large cohorts and get at least the amount of variability due to the running of different days. I know that's a mouthful, but.
So that actually is again a very good question. So the way we approached it, or typically try to do studies to reduce the variability that we have in the data due to sample collection, is that we try to collect the samples over time and then perform a single analysis using mass tag-based barcoding. And also in this study, once the blood samples were taken, the PBMCs were isolated, they were cross-linked in 1.6% PFA, and then the samples were frozen at -80 degrees Celsius. And then after a certain time point, when we thought, now we likely have enough samples collected to be able to do statistical analyses that make sense, then we barcoded all of the samples. I showed this at the beginning of the talk. And that's how we can avoid a large variability introduced by sampling over larger time frames.
Thank you so much, Dr. Bodenmiller. And again, I want to thank our audience members for these great questions. And any questions not answered today will be answered via email. We have time for one more question. Do we need follow-up biospecimens to perform such pseudo-time analysis? And if not, how do we make assumptions about what cell populations or cytokines or chemokines are decreasing or increasing over a number of days?
So again, that's a very good question. And in an ideal experiment, you would sample blood from the same patient over the course of the disease. At the time when we collected those samples, that unfortunately was not possible. We did, however, for 4 patients, sample blood at 2 different time points. And there we could actually confirm all of the trends that I've shown you in the pseudo-dynamic or pseudo-time analysis. And that gave us confidence that the assumptions that we make about the changes over time are likely correct. But I completely agree, we should have more patients. where we do sample from the same patient blood samples over the course of the disease. And if you have such samples available, we would be excited to help and to analyze them. Thank you.
Dr. Bodenmiller, thank you for your presentation today and for your important research. And I also want to thank again all the presenters, Dr. Herbrich, Dr. Davis, Dr. Bodenmiller, all of you, thank you again. And I want to thank Fluidigm for sponsoring today's event. Before we go, I also want to thank our audience members for joining us and remind you that questions submitted during the on-demand period will be addressed via the speaker, the contact information we provided at the time of registration. And this webcast can be viewed on demand through September of 2021. Until next time, take care, stay healthy, bye-bye.